Lecture 2 of Cancer Discussion intended for RCBC Spring 2020 students, but available for anyone to listen. This is another book that is really very interesting called The Immortal Life of Henrietta Lacks. Henrietta Lacks, I'm talking about here during this discussion because her cells were the very first human cells ever to be cultured outside of the body. So when I showed you a picture during the last lecture of cells that grew in a culture dish like this and how they grew on top of each other out of control, I mentioned that some of them even became detached and they will float around. And that is what happened to Henrietta Lacks's cells. There was a biopsy done. The cells were removed and evaluated, but then there was a scientist who put them into a flask and they continue to grow in there. Those cells are called HeLa cells for Henrietta Lacks. And she and her cells have impacted so much research. I myself had the privilege to grow her cells as a part of the research that I did years ago. And it is a wonderful tribute to be able, and a wonderful legacy, to be able to take a negative of her having cancer, but be able to impact the lives of so many others. A great book discussing not only the science, but her personal life as well. Okay, what happens with cancer? We looked at this cell cycle before. We talked about mitosis, and we talked about these gaps, these checkpoints, gap 1 and gap 2, that are involved in the cell cycle and what they are doing. There's a slew of proteins that are evaluating in this gap whether mitosis occurred correctly. And then after DNA is replicated in the S phase, there are another host of proteins which evaluate whether that part of the cell cycle occurred as well. Many of the mutations in cancer occur, many of the mutations are in the proteins that are part of these checkpoints, a part of these gaps. In this graphic, we can see what is a normal tissue here and here. And also you can see different cell types within this tissue. And the cancer which interrupts with this tissue is here. That's the tumor, which is just an abnormal growth of cells. Originates from this bottom layer. But you can see that the cells themselves are very different. They're kind of jagged, disorganized. Even though they're growing out of control, they're acting immature. They're not doing the job that these cells do. And they're interfering with the function possibly of this tissue. Now, tumors come in two varieties, benign and malignant. Benign is what you want to hear. Benign is a tumor is an overgrowth of cells. However, the cell type is still kind of organized. They don't break away, and that is not cancerous. When cells become cancerous or malignant is when you can see a lot of disorganization here, is when the cells begin to detach from the tumor and they then hitch a ride into blood vessels or lymphatic vessels and deposit themselves. They leave the vessel and deposit themselves in another area. That is called metastasis. What I want you to envision is that within these vessels, there are these are cells. That's one cell there, another cell there, another cell. And those cells have small gaps between them. And the gaps are how these tumor cells get into the vessel and also leave the vessel. This metastasis results because cancer cells don't care about anchorage dependence. Remember, we said that's what normal cells do. They want to attach to each other or to a surface. Cancerous cells don't care about that. In this slide, we want to take a look at different cancer types and the risk factors or carcinogens that are associated with those cancer types. Carcinogens are mutagens. They cause a mutation, but specifically they mutate genes that will lead to cancer. Breast, prostate, and lung cancer are the most prevalent cancers in the US. And we can take a look over here at the different types of risk factors. You don't need to memorize all those, but hormones, oral contraceptives contain hormone. Also, diet, obesity, smoking, of course, for lung cancer, but also prostate and breast. In some cancer types, your ancestry, your ethnicity may provide an additional risk. We can also take a look here at colorectal, which we'll discuss in a minute, and diet plays a role in colorectal cancer. Things like UV light, also other types of radiation like X-rays. These are two different types of viruses. This is a bacterium, another virus. Smoking, we see that show up again and again. And the reason I show you these is that, one, we want to try to avoid these risk factors, right? We already might have certain mutations which are going to predispose us to a cancer. So why add an additional risk by participating in these activities? But also it shows us how we can potentially cure these cancers if we understand what causes them. The next slide shows us those types of cancers again here, but in this case we're looking at the total number of new cases for that cancer. And in this case, it might be, this is a little bit old now, but you can see that the new cases versus the deaths in certain cancer types, you have a much better chance of getting treated and cured of that cancer than in other cancer types like lung cancer. And then you have cancers like pancreatic cancer, where the deaths and the cases are very close in number. And there are multiple reasons why this is the case. Certainly, when we can see a cancer and diagnose a cancer early on, then we're able to catch it. Things like breast cancer, right? You can do exams and you can feel a lump and then catch it in an earlier stage. Whereas maybe pancreatic cancer, we cannot, and you often don't feel pain, so there wouldn't be a reason to think that there was a problem. And when you eventually do know that there is a problem, it is too far along for us to treat it in a successful manner. The next slide gives us a sense of different cancer types within the world. And these aren't the top cancers in these different countries, but it's sort of the outlier, meaning how the US has colon cancer. Why is it that we have a higher incidence of colon cancer than other people, right? Brazil has a higher incidence of cervical cancer than other countries. That's really what this is showing here. And what this allows us to do by these demographic studies is to figure out what the risk factors are that are associated with those cancer types and learn that based on the lifestyle of the people that live in certain countries. And that's where that table, one of the places that the table that we saw a couple slides ago can be drawn up because of studies like this. This illustrates and drives the point home several points. One, cancer is a genetic disease. You can always find messed up DNA or modified DNA, like maybe a methyl group is on it and it's silencing the DNA. But it also drives the point home that it takes several different mutations for cancer to ensue. Let's just say we are this organism right here. We only have 4 chromosomes, and whatever perfect is, I'm perfect right here, right? I don't have any genes that are mutated at this point. And then you can look down here at the end where I have 4 different genes that are mutated, indicated by these red lines. And that's when the cells become malignant. But you can see that in between, I have other mutations that pop up because, bam, maybe UV light or smoking or diet. But I still have normal cells. They're starting to look a little bit different, but they're not cancerous yet. And the reason that is because those checkpoint proteins, those gap proteins that I told you are checking to make sure things are okay, you have a lot of those proteins. There's redundancy in those proteins. And so when one of them gets hit, we have 3 others that are doing the job. When 2 gets hit, you have 2 others. And eventually you just break too many of those proteins and we can't control the cell anymore. So cancer is genetic. It does not necessarily mean it's inherited. So you can be born perfect like we saw in this description here, but maybe not. Maybe you're not aware of it, but you are born with 2 different mutations. And that means that you are a little bit closer to developing a malignant cell because now you only have 2 different genes that need to be mutated in the course of your lifetime. That is where we talk about predisposition. And when you go to the doctor and they say, has anybody in your family had breast cancer or prostate cancer? They're trying to figure out whether you possibly might be somewhere here as opposed to here. And why is that good to know? Because we can then eliminate the risk factors associated with different cancer types because we already know that we have this intrinsic risk factor within our genes. The last slide I want to talk to you about here is how cancer works. There's 2 ways. And I want to just use the car analogy that we have a gas pedal and we have a brake pedal. But what I want you now to imagine is that in our body, we have many gas pedals and many, many brake pedals. And so those pedals are genes. They make proteins. And those proteins are either going to accelerate the cell division or they're going to inhibit cell division. And many different genes can be a part of the gas pedal or can be a part of the brake pedal. But they are all about either a gas pedal being stuck down in the down position and the on position or a brake pedal that's just not responding. We will pick up in the next lecture with specific gene types and gene names that are associated with cancers and what their job is within the cell. And that will be part 3. Thank you very much.